Hello everyone. I am sure that you have heard of brain scans and possibly even have had one yourself. But have you ever wondered about how brain scans actually work? How do we get those neat pictures of the brain? That is the topic of today's mini lecture. More specifically, drawing from Chapter 3, we will be looking at what a brain scan is, and the different types of brain imaging techniques or scans like EEGs, PET scans, and fMRIs. Brain imaging techniques or brain scans are tools for mapping brain structure and function. They achieve this through a variety of mechanisms, as we shall see with regard to EEGs or electroencephalograms, PET scans or positron emission tomography, and fMRIs or functional magnetic resonance imaging. Let's start by looking at the electroencephalogram. EEG is a non-invasive technique that uses electrodes placed on the scalp to measure brain activity, more specifically postsynaptic potentials, EPSPs and IPSPs, in the cerebral cortex. In this way, and like other brain scans, EEGs act as neural correlates of specific brain functions. For example, during the performance of a cognitive task like reading a sentence on a computer screen, large populations of neurons are synchronously depolarized, leading to a spread of electrical current throughout the scalp. This results in tiny differences in voltage measured in microvolts, recorded by scalp electrodes as shown here, which are then averaged by a computer into what we commonly refer to as brain waves. In addition to neurocognitive research, EEGs are widely used in the diagnosis of conditions like epilepsy, as well as in sleep research. Some advantages of using EEGs is that they have great temporal resolution in the order of tens of milliseconds, meaning that they capture brain activity very fast, almost as it unfolds. They are also non-invasive, meaning you don't need to penetrate the body in order to get a signal. Some disadvantages are that EEGs have quite low spatial resolution, meaning that we can't pinpoint with any precise accuracy where the signal is on the scalp or even where it's coming from in terms of its source in the brain, known as the inverse problem. What types of brain waves might you see on an EEG? Any one of these 5 basic types, which are characterized by their frequency in hertz or cycles per second. For example, the highest frequency brain waves are gamma waves at about 35 hertz or more, and are representative of mental effort. In contrast, the delta band at about 0.5 to 4 hertz is characterized by low frequency waves typical of deep sleep. How about positron emission tomography or PET scans? Unlike EEGs, which measure electrical activity of the brain, PET scans instead measure neuronal activity as a function of oxygen and glucose metabolism. They achieve this by tracing radioactive glucose during a given psychological task. Basically, a tag or a radioactive tracer like fluoride is attached to glucose, which, as we know, is brain food, and subsequently injected into a person's bloodstream. As glucose is used up by the brain, the tracer decays and emits positrons, or electron antimatter, which collide with electrons, thus producing gamma rays. These are what's picked up by detectors around the head, in essence signaling the distribution of glucose uptake in the brain as it performs a task, like seeing or hearing a word, as is shown here. Because they measure energy consumption in the brain, PET scans are used in cancer diagnosis since malignant tumors are typically highly metabolic and therefore rapidly synthesize radioactive glucose. Some advantages of using PET scans is that they are a direct measure of brain metabolism, but they have many disadvantages, including the fact that they're very invasive. They use ionizing radiation. They have limited spatial resolution in the order of 5 to 10 cubic millimeters, and they have quite poor temporal resolution, up to 30 or 40 minutes. How about those pretty colorful fMRI pictures? How do we get those? Functional magnetic resonance imaging is the more active cousin of the regular MRI, which you can get on any part of your body. The difference lies in the fact that, as signaled by the lowercase f in fMRI, this type of scan measures brain function rather than mere structural anatomy. How do fMRIs work? They measure regional cerebral blood flow and oxygen metabolism through the magnetic properties of blood, or more specifically, hemoglobin, the oxygen-carrying protein in your red blood cells. This is termed the bold, capital letters, blood oxygen level dependent contrast. Basically, by using a giant and extremely powerful magnet, just as an indication, a 3 Tesla magnet is the equivalent of about a 60,000 times a magnetic field. It's the equivalent of generates the equivalent of a magnetic field that's about 60,000 times greater than Earth's own magnetic field. So that's really powerful. By using this giant magnet, the fMRI scanner generates a magnetic field to align hydrogen nuclei and produce the images. Specifically, during a given task like tapping a finger, brain areas responsible for finger movement will need increased blood flow and oxygen consumption, thus resulting in changes in the ratio of oxygenated versus deoxygenated hemoglobin, which have different magnetic properties. Thus, this in turn leads to an increase in coherent spin of hydrogen protons that are picked up by coils in the magnetic resonance scanner, allowing for the building of a functional brain map indicating the precise location of increased neural activity. I know, lots of physics. Some advantages of using fMRIs is that they use non-ionizing radiation. They have high spatial resolution in the order of about 3 cubic millimeters, and relatively high temporal resolution in the order of a few seconds. So not as high as EEGs, but certainly higher than PET scans. Some disadvantages are that they are quite costly. There are some safety concerns if somebody has metal objects in their body or on their bodies, like pacemakers or metal jewelry. They are indirect correlates of metabolic activity and brain functions, unlike, say, EEGs, which are direct correlates of those functions. As a comparison, then, you can see here on the left an image of a PET scan showing computer-generated colored areas of high metabolic or glucose activity. In contrast, the structural MRI image shown here in the middle is a black and white static image of the brain. Lastly, the functional MRI shows colored areas overlaid on top of a structural MRI. These represent areas of high oxygen consumption during a specific task. Finally, in addition to EEGs, PETs, and fMRIs, there are brain imaging methods that focus on other aspects of brain structure and function. As an example, diffusion tensor imaging, or DTI, is an MRI technique that, unlike fMRI, which measures oxygen changes in blood flow, it focuses instead on measuring the direction of motion of water molecules within axon tracts across the brain, what we call white matter. Therefore, DTI reveals the organization of axon fibers that connect different brain areas. As shown here in color-coded axon bundles, DTI is also very sensitive to the detection of neurological disorders like Alzheimer's and MS. So to conclude this mini lecture, as objectives at the end of this lecture, you should be able to compare and contrast the mechanisms of different types of brain imaging techniques and explain the use and advantages of different types of brain imaging techniques. That's it for today, everyone. Take care.